So typically I begin my shift doing quality control and maintenance on the machines. Here I am grabbing the calibrator required so that I can go ahead and get started on this process. Here I'm setting up my calibrator so I can go ahead and load it into the machine and begin calibration. After calibration and maintenance has been done, I can begin processing and receiving samples. So here I am receiving a sample that was sent to the lab through the tube system. I'm going to go ahead and grab the specimen, and then I'm going to go ahead and begin scanning it to mark it as received in the computer system. A lot of times, specimen that is sent to the lab is no good. Either it's short or hemolyzed. And when that happens, it is my job to reorder it. And after I reorder it, I call the phlebotomist to let them know that the lab is requesting a redraw. Once I have marked the specimen as received, I'll go ahead and begin to centrifuge it, which means load it into this machine. And the point of this machine is to spin it to separate the red blood cells from the serum or the plasma. Here is the machine, and it's about done. We spin it for 5 minutes. That's what it looks like afterwards. And so now we can use that serum or plasma to run the tests ordered. And so here's me loading it into the machine. Another key aspect of my job is working in the microbiology department. Here I am analyzing temperature to make sure that everything is stable. I'm also making sure that the department is stocked. Anything that I see running low, I go ahead and go restock it. So here I am in the walk-in fridge grabbing some more gel plates. After everything has been restocked, I can go ahead and begin to culture a specimen as they come through. Here I am performing a 4-quadrant isolation on gel plates. You get these culture loops, you plate the specimen, and the goal is to isolate the bacteria so that the CLS can analyze it further. And that's what it will look like if done correctly. Other tests that are performed in microbiology can include COVID flu test, MRSA test, and various other cultures. After I plate the specimen, I can go ahead and place it into the incubator, which I'm doing here. That encourages growth, which the CLS will see the following day. I also plate things on gram stains, which are thin glass slides, and then we dye them, which is what I'm doing here. I'm staining it. And this is what the COVID flu test looks like after we prepare it. We go ahead and load it into the PCR machine, which takes about 45 minutes to complete. All wrapped up. I just finished working 10 p.m. to 6:30 a.m. Nice sunny morning now. And time to go home. Thanks for joining me.